I'm going to mention a little bit about evolution of the X and the Y in terms of how it affects fertility. But I thought I'd first start out, as requested, with a little funny slide there. Wait a second. Oh, I got off that slide. Oh, I'm sorry. Well, I wanted to show that slide, but I pressed the wrong button too soon and I can't go backwards. But it was a man who was coming in from fishing, and his wife was sitting on the couch looking kind of surprised. And he came in from fishing. And, you know, we know that men and women are different, and we just don't know why. And he says to his wife, why can't you be more like a mountain stream overflowing with trout? And she's looking at him quite surprised. So that was a funny New Yorker cartoon that emphasizes that we've all been fascinated for all our lives with the difference between the sexes. And the X and the Y chromosome really has a message for that. But I'm going to emphasize the very clinical aspect. So as Jenny was saying, we all started out the X and the Y as a pair of ordinary autosomes about 300 million years ago. Oh, wait, I'm not advancing. Something's wrong. Maybe someone help. I'm not. It's not advancing.
I would unshare and then share again. That'll work.
I'll share again. Screen share. I'm sorry about that. Okay, it's working now. And we just can show that over the course of time, once there was the evolution of a SRY gene, or rather a sex-determining gene, it was only of any function if it didn't recombine with its autosomal partner. And that was the origin of sex determination, chromosomal sex determination. Now, the default mechanism of sex determination really is temperature, the temperature in which the eggs are incubated. But in some animals, such as mammals, this SRY gene developed. In birds also, XX system developed, and they were able to evolve from the dinosaurs. So sex-specific, sex-determining genes develop over and over again, and they're really all different. But at least for our mammalian Y, eventually we wound up with a, as Jenny would say, a pitiful little Y chromosome, but maybe not so pitiful. The appearance of the male sex-determining gene on a non-recombining locus, that was crucial, of one of what was an X chromosome before, 300 million years ago when we were just reptiles. Subsequent evolution of mammals occurred after that, and that resulted in a generation of the developing Y because of the failure of recombination. Recombination is actually where chromosomes, routine mutations and deletions that always occur can be corrected by that process of homologous recombination. When you no longer have meiosis or recombination, the chromosomes always deteriorate and degenerate. And so that's happened in various animals, and it's resulted in the X chromosome being very well conserved, even though the Y is widely divergent. So there's really not much similarity between the Y chromosome on humans and even chimps and gorillas, let alone on mice or on Drosophila, or on worms. There's really, the only similarity is the Y is short and seems to determine that it's going to be a male, and the X is bigger. And if it's XX, it's a female, and if it's XY, it's a male. But the genes are totally different. This evolved over and over and over again in different species. The ancestral Y underwent no meiotic recombination around the SRY gene. The ancestral X did undergo meiotic recombination in the female, however, and therefore the X is extremely well conserved in all mammals, whereas the Y is widely divergent. But remember that the X chromosome spends 1/3 of its evolutionary life in the testicle, where it's not recombining. That means that there are some parts of the X chromosome that are very similar to the Y. We'll get into that later, but it has to do in an important way with how the X chromosome also is very important for fertility in the male as well as the female. Now, it also relates to the non-hormonal differences between men and women. While the X chromosome remains stable, the homologous genes on its mate, really, its mate was the Y, were decaying. At the same time, new genes which control spermatogenesis arrived on the decaying Y. Well, that's because transpositions occur all the time. Genes will jump from one chromosome to another all the time, and it's probably the origin of speciation, in fact. But whenever a spermatogenesis gene arrived on the Y chromosome, it stuck there because it was a safe harbor. Once on the Y, these formerly single copy genes, autosomal genes, amplified into multiple copies and achieved greater prominence. So why is that important? I'll show you later that the DAS gene, which we're all now familiar with, the most famous male infertility gene, has 4 copies on the Y. And but there is one rescue copy on chromosome 3. And in fact, the DAS gene originated on chromosome 3 as what we call DASL. And when it was transposed in the monkeys to the Y, it amplified and became much more important than the original DASL. Nonetheless, in Y chromosome deleted men with AZFC deletion, you'll almost always find some sperm because chromosome 3 has a DAS homolog. So we have delay of ancestral autosomal genes on the heterogametic sex chromosome and parallel accumulation of genes that control spermatogenesis. Because as Jenny said, I mean, the Y would have completely disappeared, except that we have this accumulation of genes from all over the genome that controls spermatogenesis. Now, X inactivation is a very important subject because when these genes decay on the Y, so you would think that women have 2 copies because they have 2 X chromosomes of these intelligence genes and other genes, and men only have 1 copy that's functional. You would imagine that, women would be twice as intelligent as men. Now, I know they're more intelligent, but they're not twice as intelligent. So, in fact, men would be dead if it weren't for some mechanism to give us equal expression of X genes. So over time, the ancestral Y shrunk because of decay of X homologous genes. But the loss of those ancestral Y genes were compensated for by hyperexpression of Y homologs on the X and X inactivation. In other words, those genes on the X that had decayed on the Y become hyperexpressed so that in the male, at least, he has as much expression of those X genes as the female. On the other hand, you'd have too much X expression and the female couldn't survive if there wasn't X inactivation, where half of the cells in the body actually have the X chromosome inactivated, so there's no expression. There are genes that escape X inactivation, and we're going to point out later that that is probably one of the most important reasons why there are non-hormonal differences between men and women. For non-hormonal differences being like autism, more common in males, or like autoimmune disease, more common in females. So this way, males did not suffer from loss of genes, but females did not have twice as many functioning X genes as the males, and so we were all relatively equal. And this is just a diagram from Jacalian and Page paper on this in 1998, showing this is a gradual process. First, you developed a pseudo-autosomal gene, and the X-linked gene was not X inactivated, and the Y-linked gene had a reduced expression, and the X had increased expression, and then eventually you had to have X inactivation so that both the male and the female would have equivalent expression of what were extremely important X genes, which were really autosomal genes. So we know that for years we just looked at the karyotype and we thought the Y chromosome had nothing to do with anything except sex determination. But then when we did our first study in 1995, just mapping the Y chromosome, and it was the first mapping of any chromosome, we only picked the Y because it was short, and we thought that it might have something to do with spermatogenesis. As it turns out, it's probably the most difficult chromosome in the whole genome to sequence because of multiple repeat copies. So anyway, so there were no deletions detected in 90 fertile males that were azoospermic. But we detected these deletions in what we call the AZFC region in 13% of azoospermic males. And that was the origin of the AZFC deletion. However, Then we got in much more detail because this mapping program was maybe 1 every 30,000 base pairs. So, I mean, you're like trying to find a needle in a haystack. And when we looked at it in more detail with AZFA, AZFB, and AZFC regions, still not enough detail, we were able to find that the common deletion was in this AZFC region, which resulted in a combination of maturation arrest and Sertoli cell only. There were often some sperm located. And when we did more detailed test studies, what we found was in this, if you had an AZFC deletion, there were almost always some sperm, either in the ejaculate with an extremely low count, or in the testes if you did a difficult microsurgical testa procedure. And where did all this happen? Well, the original insight to hybridization studies showed the DAS transcripts in the spermatogonia, which explains why DAS is probably the most important original spermatogenesis or even oogenesis gene, but also why you could have a combination of maturation and Sertoli only because it's expressed in spermatogonia. Now, as I said before, there's no relationship to any of the Y chromosomes in Drosophila or worms or even fish or even mammals. They only are called Y because they're smaller than their sex chromosomal counterpart, but they're all different. And so DAS is found in the Y chromosome only in humans and in higher primates because there was a transposition from chromosome 3 to the Y between the transition between old world monkeys and new world monkeys. And DASL is found on chromosome 17 of the mouse, but not on the Y. And the mouse has a gigantic Y chromosome. DASL homolog found on chromosome 3 of humans, 1 copy, but DAS is found in multiple copies in the Y. So the DAS homolog, it's just like DAS, the Boole gene it's called, is found on an autosome of Drosophila, but not on the Drosophila's Y. So it's all very confusing, except that evolution finds a way over and over again of promoting a genetic sex determining system rather than a temperature dependent sex determining system. But also from a clinical point of view, this shows why in men with AZFC deletion, that's the very best prognosis for azoospermic men. And when we sequenced the whole AZFC region, we understood what the problem was. In this region we call B1 and B2, there were 229,000 identical base pairs. And between them were 19 transcription units, 7 families of genes, including DAS. And there was a whole palindromic complex that actually exceeded that B2B2 pair, in which there were 27 transcription units, 11 families of genes that included RBMY. So we finally found out the reason why these DAS deletions, why these AZFC deletions occur, because illegitimate homologous combination, I'll discuss in a section, is where these B2, B1 genes recombine with each other and the middle drops out. So what's the mechanism of de novo appearance of these deletions? It's an inherent result of the structure of the Y. Now there's the Y chromosome has absorbed all these genes and amplified them that helps increase spermatogenesis, but then spermatogenesis is dependent on the Y. And the Y, as Jenny said, is very fragile. So what about the sequence of AZFC? We call it illegitimate homologous recombination. If you look at them, you can see that these recombination targets are identical, and what's in between them will disappear when they recombine. Now, all chromosomes must recombine to maintain themselves over time. It's a little bit of a funny slide. Autosomes repair themselves by recombining with each other during meiosis. Okay, everybody knows that. The Y tries to repair itself by recombining with itself. The DNA between identical sequence of nucleotides drops out when homologous sequences on the same chromosome recombine. And this is just a diagram of how the DAS gene was transposed from chromosome 3 in humans to the Y in humans. And this is just showing that chromosome 3 has a DAS-like or DASL gene, which is a DAS homolog that rescues spermatogenesis in non-obstructive azoospermia. Now, what about the offspring? Are the offspring of men who have ICSI that have a Y deletion, are they going to have an amplified Y deletion? Is it going to get worse? Or will they have no Y deletion? Well, it's obvious that they have exactly the same Y deletion as their father. But where did it all come from? The father of AZFC deleted men, they're not Y deleted and they're obviously fertile, but the son is Y deleted. Another example, father of AZFC deleted man is not Y deleted. And he obviously had to have good spermatogenesis because he had all these children, including a son. And the son was AZFC Y deleted. One boy from Tessa ICSI blood was AZFC Y deleted. So Y deletion occurs de novo in the fertile father's testes. And clearly the father and brother here were obviously fertile, but this Y deleted occurrence was something that happened within accidentally. The Y deleted sperm from his father, and it's probably 1 in 2,000 sperm, and every man with normal spermatogenesis has a Y deletion or Y dropout. If that's the lucky sperm that fertilizes mom's eggs, then the baby will be Y deleted and will be infertile. And so there's no transmission of the Y deletion, no increase in transmission of a more expanded deletion in the son of AZFC mutant men, but they will have the same AZFC deletion as their father. So why does the Y not completely disappear? Well, because autosomes repair themselves by recombining with each other during meiosis. The Y tries to repair itself by recombining with itself. And this is the funny part of it. Autosomes have sex with each other. The Y masturbates. And that's my cumulative way of trying to think about it. And this is a picture from the Howard Hughes Institute, where we see that what happens is because you have so many identical repeats, the Y tries to repair itself. It's not that efficient, but it's not as efficient as meiosis, but it tries to repair an area of the Y that was injured by mutation just by gene transfer. And that's one reason why the Y has held up over all these years. And it may be an argument against Jenny's postulate that the Y will eventually disappear. So this also explains multiple copies of Y genes. The Y is so difficult to sequence because of these multiple arrays of repeat sequence, which we call amplicons. Once DAS transposed from chromosome 3 to the Y, it amplified into multiple copies, but the original homolog of DAS remains on chromosome 3 as a backup. And now the complete sequence of the Y chromosome was almost impossible to do because there are so many of these ampliconic repeats, and really nobody else has even done it. And we need to be able to have other people sequence amplicons because I think there's tremendous dark DNA in these amplicons that'll explain a lot of male-female differences. So we had to have, with these huge palindromes of mirror image identical inverted repeats, we had to look for minute variants and otherwise identical repeats used as sequence family variants, and we had to do it in just one man's Y chromosome. So what we found was not only are there these multiple amplificated repeats, but there's also palindromes. What is a palindrome? Palindrome is where one side or the other side of the center point is identical in reverse order. And this is a perfect example of a palindrome. Madam, I am Adam. If you look at the center, you'll see that the either side is actually identical, but in reverse order. Madam, I am Madam. And so that's what is composed of most of this ampliconic region are not only sequence repeats, but sequence inversions as well. And those sequence inversions are another way that we can just lose huge areas of the Y chromosome. And that's how we lost AZFB. AZFB deletions take out a gigantic section, not just 229,000 base pairs, but a total of 4.9 million base pairs. So what about testis-specific versus ubiquitous expression that Jenny was talking about? So there's 3 different areas of the Y chromosome. There, after we sequence the whole thing, there's the X transposed, which is identical in the female and the male. And that they have to avoid X inactivation or we wouldn't be alive. There's the ampliconic region, which is huge, expressed only in the testes. And then there's the ubiquitously expressed X degenerate region, 16 genes that appear to be degenerated from the X homologs. They're different, but they're different. And that means that the X on homolog is most likely hyperexpressed. So this X degenerate region, which escapes X inactivation, is probably one of the most important areas to study to try to understand why men and women are different if we remove the hormonal differences. Smaller Y deletions encompassing fewer spermatogenesis genes, by the way, like GRGR, causes varying degrees of oligospermia instead of azoospermia. So we can summarize what the Y chromosome looks like in a simple graph with AZFB and AZFA and AZFC lesions. And we prefer to be more specific based on sequence like P1, P5, and P4, P1, and B2, B4, GRGR. And the reason for that is take a look at these GRGR deletions. That's like half of the AZFC deletion is deleted. And that means that these men will have lower sperm counts. It's even a polymorphism, the GRG lesion, for a greater risk of male infertility. So why don't human males have really low sperm counts? Well, it's sperm competition. And this is what the famous Roger Short, who was my idol, figured out, that chimpanzees have relations with each other as soon as one female goes into heat. Every male in the pack will mate with that female. So it's likely that the male will have the sperm count of the male offspring, will have the sperm count of the male that had the highest sperm count. Now, so you see that chimpanzees have gigantic testicles and just humongous sperm counts. What about the gorilla, this fantastically macho animal? There's no sperm competition. And he has maybe two or three females, and they never have a chance to mate with any other males. And look at their tiny little testicles. I mean, the gorillas really have tiny testicles because of the lack of sperm competition. And the Y chromosome in the chimpanzees is enormous. They have much more amplicons than the humans have, and the gorillas have less amplicons. So small deletions cause less severe spermatogenic defects. AZFC deletions remove 3.5 million base pairs. Sorry, I didn't mean thousand, I meant million. And AZFB deletions remove 6 and 8 million base pairs between 32 and 42 genes instead of 21 genes. So AZFB deletions so far seems to result in no spermatogenesis except maturation arrest. Smaller deletions like GRGR, however, remove only 9 genes, and they are only basically polymorphisms for lower sperm count. And this is, you see, GRGR deletions occur between identical regions within the AZFC that take out only a smaller amount of AZFC and leave half of the DAS genes intact. So our conclusions are milder degrees of oligospermia are caused by smaller deletions, which take out only some copies of male-specific genes, leaving intact back copies. And sperm can be found in tiny numbers in tessa cases if there is just one backup copy of a deleted gene. So AZFC is an example of a very good prognosis for tessa case, as well as GRGR deletions. Sperm count varies with gene copy number, and testa-specific genes on the Y always have multiple near identical copies. Small deletions are more common than larger deletions, and that makes sense. And these more common deletions leave backup spermatogenesis genes that result in lesser degrees of spermatogenic defect. So we can look at a graph of a bell-shaped curve of a normal population with sperm count and of a population that's AZFC deleted. And they're going to have sperm. And then the GRGR deleted group falls in between AZF deleted and a normal population. So we think that this is going to be the way we have to look for the genetics of azoospermia and oligospermia throughout the rest of the genome, but we've been stymied in the Y chromosome to find anything more. Turner syndrome is fascinating. I won't go into it in detail, but what if the whole chromosome turned into a palindrome? Well, then what you wind up having is XO is not always the absence of an X chromosome in a baby girl, but rather the absence of a Y chromosome in a baby boy. There's no maternal age effect in XO Turner syndrome. And in 3/4 of XO girls, the X chromosome is of maternal origin. So in the majority of Turner syndrome, we would guess we're not going to find any sperm because of the majority of them, and we're not going to find eggs because the majority of them are males that lack a Y chromosome rather than females that lack an X chromosome. And what about the X chromosome in general? Like the Y, the X is present as a single copy in the male. Therefore, mutations in the X that are favorable to spermatogenesis will be readily expressed in male offspring. So the X spends 1/3 of its evolutionary life in the testes where it cannot recombine, and the human genome has not been completely sequenced. We know that. And so because of all these amplicons, we do not have complete quality sequence. So I call that the dark side of the X. The X cannot escape completely some of the problems the Y solves by constructing amplicons and palindromes. In fact, 12% of the X only undergoes meiosis in the X only undergoes meiosis in the ovary, not in the testes, where it spends 1/3 of its life. So it therefore has huge ampliconic regions. 12% of the X is just like the Y. And these regions are still completely unsequenced, very likely to have a rich complement of genes for both spermatogenesis and oogenesis. But one other thing, what about the difference between the sexes? The X degenerate portion of the Y is going to be different in men than in women, especially if they escape X inactivation and hyperexpression. Autism is more common in males. Could that be from the dark side of the X? Well, actually, now we have evidence it may be really related to the Y chromosome X degenerate genes. And autoimmune disease is more common in females. Well, all this could be related to the fact that there are differences between the sexes that are related to the X and the Y chromosome, but are not hormonal. So I'm not going to go any further. My time is up, but I can answer any questions about what I think is extinction in general and how animals go extinct and why the dinosaurs went extinct. It's one of my favorite subjects. But my time is up now, so I'll close now and I'll give it back to the, I have other slides, but I think my time is up, so I'll stop with that.